Inside every cell is a black box that stores the entire code of life. Open the lid and watch DNA operating in real time. This is the nucleus, the vault of your genetic data. Chromatin fibers await unpacking to reveal the double helix. As chromatin loosens, the double helix gradually emerges. Structure determines function, the core principle of molecular biology. The DNA double helix is only 2 cm thick, rotating at Clark 10.5 base pairs per turn. A sugar phosphate backbone cradles the letters A, T, G, and C. The major and minor grooves are not merely geometry. They are the doors proteins use to recognize and read genes accurately. The negatively charged sugar phosphate backbone stabilizes the strand, shielding the core base sequence from random collisions. A pairs with T via 2 hydrogen bonds, flexible enough to open and close during replication. G pairs with C via 3 hydrogen bonds, a tighter bond whose small detail has a big impact on local stability. The human genome contains R3's 109 base pairs, a colossal number that cells manage every day. DNA's 3 duties are storage, replication, and instruction. If the cell is a factory, DNA is the technical specification. A gene is only a short stretch in a sea of sequence. Pinpointing the correct locus is prerequisite for accurate expression. A helical pitch of 10.5 bp per turn optimizes packing and accessibility. Tight enough to be compact, loose enough to be reached. A diameter of 2 nm, about $120,000 the width of a hair, means even tiny micro errors can bend the helix. With only 4 letters A, T, G, C, arranging them in triplets yields an almost limitless language. Codon to amino acid. Every 3 bases specify 1 protein building block. The genetic code is a dictionary shared by most life, from structure to function. When letters fall into place, the cell receives the correct instructions. When they don't, the repair systems step in. DNA replication begins as the fork opens like a zipper, and every step obeys the 5-3 direction. Helicase splits the strands, consuming ATP like a molecular motor, with speeds reaching hundreds of bases per second. NCBI PubMed. SSB proteins coat single strands to prevent re-annealing, like paper clips holding a page open. Primase lays RNA primers as starting pads. Without primers, polymerases cannot initiate extension. The leading strand glides 5'3", adding nucleotides continuously. The express lane of replication. The pCNA sliding clamp embraces polymerase, boosting stability and speed like a ball bearing in an engine. The lagging strand forms discrete Okazaki fragments that are joined later, an elegant design that still respects the 5-3 rule. Arnase H removes RNA primers, making room for the more durable DNA. DNA ligase welds the micronicks shut, restoring an unbroken continuous strand. 3-5 proofreading corrects errors on the fly. Error rates can fall to 10.7 to 10.9 per base copied, NIH, NCBI. Recognizing the 5-3 polarity is the key to understanding every flow of information on the DNA strand. The replication fork in full view resembles a molecular assembly line refined over millions of years of evolution. To read genes, chromatin must open. Histone acetylation loosens the fiber so proteins access the promoter more easily. The promoter, such as the Tata box, is the signpost, "Start here." Misplacement throws the transcript off beat. The pre-initiation complex assembles with TFIID, TFIIH, and others, creating a transcription bubble, like scaffolding for machines to dock. RNA polymerase II runs along the gene, synthesizing a nascent mRNA 5-3. During elongation, complementary nucleotides pair up, balancing optimal speed and accuracy. The 5 cap is a protective helmet that stabilizes mRNA and targets it to the ribosome. Splicing removes introns and joins exons. A single gene can yield multiple transcripts, the basis of diversity. The poly-A tail increases stability and regulates export and translation. The longer the tail, the more broadcast-ready the message. mRNA exits the nucleus through a nuclear pore, a gate of stringent quality control. At the ribosome, the AUG codon issues the start command, and methionine becomes the first brick. tRNA cycles APE, lengthening the polypeptide with each beat like a precision sewing machine. As the protein leaves the ribosome, it folds from secondary to tertiary levels. Correct structure dictates correct function. The environment leaves its marks. Ultraviolet light can deform DNA after just minutes of unprotected sun exposure. WHO. A thymine dimer bridges two adjacent Ts, bending the double helix and obstructing polymerase. The cell responds via nucleotide excision repair, NER.
X-ray particulate
Yet
density
high damage
high.
loads can overload the system. Cigarette smoke and vaping create bulky adducts on bases. NIH PubMed reviews report strong links to mutational burden. These adducts mispair bases, forcing checkpoints to halt the line. The biological delay is the price paid. Chronic sleep loss and stress elevate ROS.
Oxidation.
Circadian misalignment weakens nighttime repair.
8-oxo-G
7 to
mismatch.
9 hours of sleep recommended. A classic case is 8-oxo-G, oxidized guanine that readily mispairs, seeding mismatches across replication rounds. Mitochondria are endogenous ROS sources during high metabolism. Poor energy balance increases radical leakage. Taken together, UV, smoke, and ROS are three mechanistically distinct attacks that collectively push the genome toward instability. As mismatches rise, the cost of repair climbs and cellular resources are diverted from other tasks. Checkpoints trigger alarms to preserve safety, but when prolonged, they slow normal physiology. More dangerous is the double-strand break, DSB, a lesion that erases alignment markers. Cells have two roots, NHEJ, fast but error-prone, and HR, slower but precise when a template is available. When damage exceeds repair capacity, genomic instability rises, the foundation for long-term disorders. When tumor suppressor genes falter and oncogenes activate, risk grows exponentially. The arithmetic of cumulative epidemiologic risk. At the organism level, we see indirect signals like fatigue, poor skin, and persistent cough. Do not self-diagnose, but do not ignore them. In short, genomic efficiency drops and the repair burden rises. The good news is the body can recover part of that capacity. Solution number 1. Reduce UV dose with shade and UPF clothing. Cut it at the source for maximum effect. Broad-spectrum sunscreen with an appropriate SPF, reapplied as advised, helps reduce formation of new dimers. Solution number 2. Quit smoking and vaping with a 30-day plan. Each smoke-free day reduces adduct accumulation. Use behavioral support,
Sleep, 7 to
NRT,
9
and
H.
counseling hotlines. The earlier you quit, the more benefits compound over time. WHO, NIH. Solution number 3. Sleep 7 to 9 hours on a fixed schedule and limit late-night screens. Sleep is the genome's natural maintenance shift. Get 15 to 30 minutes of morning light to align your circadian clock and improve nighttime sleep quality. Solution number 4. Whole food nutrition rich in folate, B12, from leafy greens, legumes, eggs, and fish. Raw materials for nucleotide synthesis and repair. Add polyphenols from fruit and berries, and drink enough water to support redox balance and lower baseline ROS. Solution number 5. 150 minutes of weekly aerobic activity plus 2 strength sessions. Better circulation and lower stress reduce baseline ROS. Add 5 minutes of slow breathing daily and screen